What is the purpose of this technique? Recombinant proteins are significant in the biomedical world, as the genetically modified strands of protein can assist in a number of medical situations as treatment options, from human insulin in cystic fibrosis to antibodies in cancer treatment. Recombinant proteins are classified as proteins encoded by a gene produced through recombinant DNA technology. These proteins are artificially created from two or more sources into a single recombinant molecule. These genetically modified sequences would not normally be found in the genome. The purpose of these proteins is to reconfigure sequences to create products that can be beneficial in the medical world. Recombinant proteins can be utilized in the medical, pharmaceutical, and agricultural industry. Genetic engineering, like recombinant proteins, can utilize modern tools and approaches such as molecular cloning and transformation, which are less time-consuming and yield more reliable products. Currently, more than 170 recombinant proteins are produced and used in medicine worldwide. For example, epoetin alpha is a well-recognized recombinant protein that can effectively be used in curing of anemia. The history and development of recombinant proteins. The process of recombinant proteins in medicine came about by altering a sequence or code of DNA, therefore resulting in the product of recombinant proteins. These proteins have provided breakthroughs in biomedical biotechnology, being used in both biomedical research and treatment. Recombinant DNA and biotechnology has always been controversial, particularly in the debate regarding recombinant DNA, as it could lead to the resolution of many medical problems in human history, but could also be used for personal gain in the sense of improving beyond what would be considered to be normal. The first recombinant protein used in treatment was recombinant human insulin in 1982. The development in protein therapeutics is significant in the field of medicine, but the role it has is still in its infancy. Development of recombinant proteins over the years. In 1953, Dr. James Watson and Dr. Francis Crick revealed the 3D structure of DNA, which is still used today. In 1973, John Morrow and colleagues clone and propagate ribosomal DNA genes from a eukaryote in E. coli. In 1976, the NIH releases the first guidelines for recombinant DNA research, and the first recombinant expression is evident in E. coli. In 1982, advancements in biotechnology began with the creation of a biosynthetic human insulin by Herbert Boyer, and the first recombinant human insulin was developed. The U.S. Food and Drug Administration approved the first biologic or recombinant protein. In 1992, the FDA approved the first genetically engineered blood clotting factor, a recombinant protein used to treat hemophilia A. They also approved a recombinant protein to treat renal cell cancer. In 1993, the FDA approved a recombinant protein to treat multiple sclerosis, marking the first new multiple sclerosis treatment in 20 years. In 2006, the FDA approves a recombinant vaccine against human papillomavirus, which causes genital warts and can cause cervical cancer. Today, recombinant proteins are being developed for a number of things. Treating chronic pain, which is one of the top issues that can impact quality of life, can be assisted through recombinant technology. A fusion protein of the anti-inflammatory cytokines IL-4 and IL-10 can optimally treat debilitating pain conditions and regulate aberrant neuroimmune interactions. Fermentation of recombinant proteins can assist in reducing the development time for universal vaccines and create an effective control of vector-borne diseases. Prevention of chronic diseases is also a possibility, with human insulin having been one of the first proteins to be developed. Recombinant proteins have also surfaced as agents in assisting treatment and prevention of diseases and illnesses, with half-life recombinant proteins assisting in simplifying the clotting management of those with hemophilia. Extending the life of these proteins reduces infusion frequency, which may also reduce the frequency of spontaneous bleeds and potential for hemophilia complications. As stated before, recombinant technology is in the beginning stages, and obviously with technology and science rapidly advancing, it can be expected to see recombinant proteins in more products. Screening and implementation of recombinant therapeutic proteins will require high-level methods to ensure that development is safe and appropriate.
Recombinant protein and the continued development of its technology is an important technique in the field of medicine, as there are so many applications and uses for them. As with anything though, there are always drawbacks in the technology and techniques that can still be developed. With this in mind, we look at some of the main recombinant expression systems and discuss the advantages and shortcomings of each different technique. The bacterial expression system is the expression system most commonly used, as there is extensive knowledge in bacteria. The most common bacteria species used for the expression system is usually Escherichia coli. So we'll just quickly go over how this expression system works. So using restriction enzyme isolates the gene of interest. That gene of interest is then introduced through a vector, and usually with the bacterial expression system, plasmids are commonly used. So once the plasmids have the gene of interest combined with its own DNA, it is then introduced to the bacteria for uptake. The application of markers will help determine which bacterial cells have taken the gene of interest in and which have not. Markers in this case usually being antibiotic-resistant genes. With this marker, determining which bacterial cells have taken up the gene of interest and which have not is simple, as bacterial cells which have taken up the gene of interest and additionally that antibiotic marker will continue to grow, and those without will obviously not. With the proper nutrient supply, these bacterial cells will continue to replicate, and with this, the replication of the gene of interest. So what are the advantages of the expression system? First of all, bacterial genetics are easy to manipulate and only require the use of common laboratory equipment to do so. The ability to do this allows scientists to influence things like growth rate to life expectancy of the cells. The wealth of knowledge in bacterial genetics, genome sequencing, and physiology is a desirable trait, as it eliminates the amount of unknown variables, which is such a helpful trait when it comes to the construction of recombinant proteins. Bacteria, being a prokaryotic cell, are a lot simpler in their ability to express compared to eukaryotic cells. It is one of the reasons it has the ability to rapidly express recombinant proteins, having a significantly shorter doubling time compared to eukaryotic cells. The overall expense of developing recombinant proteins is less expensive compared to a lot of other systems. This being because the media required culturing the bacteria can be produced from components which are readily available, making them inexpensive. There are also a good amount of vectors and host strains that have been developed to maximize expansion, making it very effective when comparing the ratio of cost to product produced. So there are a number of shortcomings for this expression system as well. Because bacterial cells are prokaryotic cells, it lacks and is limited by its post-translational modification abilities, having an effect on glycosylation, formation of disulfide bonds, phosphorylation, and proteolytic processing. All this meaning that there is more or less a probability that the expressed protein from this system do not fold properly and therefore make them not bioactive or functional for the application in medicine. Because of the difference in cells, prokaryotic to eukaryotic, there is a difference in codon bias. What this means is that most amino acids have been encoded with more than one codon, and all the available amino acid codons are bias utilized by different organisms. So this bias sequence used for an amino acid will be favored differently in the prokaryotic cells to eukaryotic cells. This factor essentially impedes gene expression and protein synthesis. Toxicity can also be a major problem because usually when trying to synthesize a recombinant protein, that protein usually is toxic to the bacterial host cell used to synthesize the protein, and this usually prevents the cell cultures from reaching its maximal densities. The mammalian expression system would be the most suitable from a medical application point of view, most identical to cells for that of human application. There are two methods to the system. You can either introduce the gene of interest in mammalian cells through viral infection, where the gene of interest is inserted into a virus, and it's the virus that transports the gene of interest into the mammalian cell. The second method does not use any vector at all, and the gene of interest is inserted into mammalian cells directly through microinjections and electroporation techniques. The most commonly used cell lines for the expression system is usually Chinese hamster ovary cells, baby hamster kidney cells, melanoma cells, and human embryo kidney 293 cells. So just a quick overview on how the system works again. We isolate the gene of interest by using restricting enzymes, and the gene of interest gets inserted into a virus, and that virus then being infected into mammalian cells. Or we use the alternative method where the gene of interest is inserted directly into mammalian cells by microinjections and electroporation. Once mammalian cells have been introduced to the gene of interest, it begins to be expressed and replicated. So the advantages of using mammalian cells is that the post-translational modification in these cells are very similar to how these processes would occur naturally in the human body, in their most natural state in terms of their native tertiary structure, physiochemical characteristics, and bioactivity. Its ability to perform complex post-translational modifications ensures the synthesized proteins' efficient secretion, drug efficiency, and its stability. Its ability to perform the proper folding as well of the synthesized protein is also of a great benefit, as misfolding and also incorrect post-translational modifications can lead to undesirable immune responses. Mammalian cells lines are also very adaptable to changing environment conditions during the production stages, having a very large window for changes. They are more flexible to changes in temperature, oxygen levels, pH changes, and pressure changes, making them very easy to handle. Unfortunately, the mammalian expression system is associated with being very expensive to operate, as it is a complex system to operate and maintain. Therefore, it requires a specialized workforce to be specifically trained. And in addition to this, the media required for the expression system is quite expensive as well. It is also quite difficult to optimize concentrations for cell growth, and for the amount and quality of the product that is to be produced. This being important because for the cell lines to achieve its full potential, it is important to create a suitable environment that provides the best chemical and physical conditions. It is difficult to optimize concentrations because it is estimated that over 50 various components are required. With mammalian cell lines, they are very susceptible to microcontamination, and this is usually a common problem. With the cell lines being rich in nutrients such as glucose, ammonia, trace elements, and micronutrients, it provides the perfect breeding ground for microorganism growth. This affecting the cell lines by inhibiting culture growth, compromising the cell line's reproduction ability. Mammalian cells are also very susceptible to viral infections as well, which have the same inhibiting effects of the cell lines as just was mentioned. The maintenance and continuous nutritional supply to the cell lines can also have a toxic effect on the cells themselves through the accumulation of catabolites such as ammonium and lactate. The main nutrients in the development of these catabolites being glutamine and glucose, which are rich in mammalian cell lines. Catabolites having demonstrated to obstruct mammalian cell growth and production. The yeast expression system is growing in popularity as a system to be used in the production of recombinant proteins. Saccharomyces cerevisiae is the most common and popular organism used in this expression system, but there are others, such as Pichia pastoris, also known as Comagatella puffy, Clivoromyces latus, and Yarroria lepeleticia. So again, we'll have a simple run-through of how the system works. It's very similar to the bacterial expression system, where we get the gene of interest by using restriction enzymes, which then gets inserted into plasmids along with markers. The plasmids are taken up by the yeast, and the yeast, with a nutritional supply, begins to grow and replicate the gene of interest. So the real benefit of using the yeast expression system is because it has features of both the prokaryotic and eukaryotic systems, which is a huge benefit when creating recombinant proteins. So the benefits of having prokaryotic features enable it to be cost-effective, enable simple genetic manipulations, high growth rates, and high cell densities. Having eukaryotic features enable it to have post-translational modifications that are more similar to that if it was produced naturally. There is also an abundance of strong promoters available for this expression system. TEF, G3P, ICL1, POT1, and GIP, to name a few. Having strong promoters enable the increased secretion of the protein being produced. Unfortunately, there is a downside to having promoters that are very strong. So even though these strong promoters enable increased cell densities and thus increased overall yield, the increased amount of transcription of the gene of interest taking place increases the stress levels in the cell, where it can get to a point which activates the unfolded protein response. This is where stress levels are so much where it gets to a point that results in the death of the cell. So there has to be a balance when synthesizing recombinant proteins using the system, where you want the promoter to be strong enough to produce a good yield. They're not strong enough, it causes the death of the cell and the gene of interest can no longer be reproduced. There are several steps when the protein is ready to be converted to become bioactive. And with the yeast expression system, there are some limiting factors when it comes to its post-translational modification capabilities. And usually the glycosylation pattern, where it significantly differs to that usually seen in the human body. This having effect on the synthesis protein's bioactive capabilities. The final shortcoming discussed here is that the system requires fermentation to produce high cell densities. Unfortunately, high cell densities cultivations of yeast through fermentation often leads to the production of increased contaminated proteins. The final expression system that we will discuss is the insect expression system, which is usually done in conjunction with the baculovirus expression vector system. The baculovirus is a family of DNA viruses that are known to affect the insect population. There are a few cells used in this expression system, the most common being derived from Spodoptera frugipruda, cell lines SF9 and SF21, and the Trochoplazia Ni cells. Other cells used are Drosophila melanogaster and Orthographa californica. So the insect expression system starts off like all other systems. Isolate the gene of interest using restriction enzymes. The gene of interest is then placed into an expression vector along with a marker, promoters, and everything that has already been discussed. The expression vector in this case being the baculovirus. Thereafter, the process requires two more steps. Firstly, the insect cells are produced to the required concentrations, and the second, that the concentrations of the cells are then infected with the modified baculovirus containing the gene of interest. The advantages in using insect cells as an expression system is that they are fast growing and higher in density compared to mammalian cells, so it's good for developing proteins where a high amount of volume is required. The other advantage of using insect cells is that there is no risk of contamination by prions. This is where the protein can trigger normal proteins in the brain to fold abnormally, and there is also no risk of contamination from oncogenic DNA, which are genes that have the potential to cause the development of tumors. So the disadvantages with this system is its post-translational modification capabilities. Just like the yeast expression system, the insect expression system struggles to mimic the glycosylation pattern seen in mammalian cells. if the protein being developed requires complex post-translational modifications, the cells in insects not being able to perform glycosylation at the endometal. The other shortcoming associated with this expression system is the same with most other systems as well, that there is usually a high cost associated when synthesizing a protein using this expression system. The culture media required to develop insect cells being very expensive, where it has been estimated to cost about €700 for 1 gram of culture media for insect cells. Put this into perspective, to properly develop and express a gene of interest using this expression system, you are going to need a significant amount, possibly in the range of liters in weight of cultured media.
Current applications of recombinant protein has grown exponentially in the last 40 years and will continue to grow as new methods and strains are researched and laboratories uncover new ways to implement recombinant protein. Recombinant protein is widely used in research, biotechnology and medicine, with research being the most common application. They are used as reagents in the laboratory to generate antibody probes to examine cells and the protein synthesis within. In the human medical world, recombinant proteins are used in a many manner of ways to treat endocrine and metabolic disorders such as diabetes mellitus, haemophilia or hepatitis B, to name a few. For augmenting hematological endocrine pathways and immune responses by binding to specific molecules to guide the immune system to destroy it. Inflammatory diseases such as rheumatoid arthritis or people suffering renal failure and or dialysis. To treat or modify diseases such as infectious diseases like HIV/AIDS and respiratory tract infections. Vaccines to protect against infectious diseases or toxins such as polio, measles and hepatitis B, and treat against autoimmune issues such as the anti-rhesus D drug for pregnant women with a different blood type to their baby. Monoclonal antibody technology is used to stimulate a signaling pathway interference or set a target for destruction on the molecule or organism, and they are an important research tool. Leader, Bakker and Golan devised a classification for therapeutic proteins in groups as shown here in this table and as discussed on this previous slides. They are divided into Group 1, therapeutic proteins with enzymatic or regulatory activity, with subgroups for replacing a protein that is deficient or abnormal, augmenting an existing pathway, and providing a novel function or activity. Group 2 is therapeutic proteins with special targeting activity, with subgroups for interfering with molecule or organism and delivering other compounds or proteins. Group 3 is devised with therapeutic proteins or vaccines, with subgroups of protecting against a deleterious foreign agent, treating an autoimmune disease and treating cancer. And Group 4 is therapeutic proteins as diagnostics, with focus on purified and recombinant proteins used for medical diagnostics. Now let's take a closer look at some of the medical uses of recombinant protein. The first treatment using recombinant protein to be approved for human use was in 1982 by the US Food and Drugs Administration, and that was insulin. This is a Group 1 therapeutic protein replacing a protein that is deficient or abnormal due to the insulin hormone not supporting the body properly. Insulin is a human hormone in which it controls glucose metabolism regulation. A person with diabetes mellitus has deficient insulin, which causes hyperglycemia. To treat recombinant human insulin is administered in times of hyperglycemia to decrease glucose levels. Recombinant human insulin is made in the laboratory by extracting the human insulin gene from the beta cells of the pancreatic islets using an enzyme such as trypsin to cleave. A plasmid vector is removed from the prokaryote E. coli cell, and the cleaved insulin gene is then ligated into the expression plasmid vector. Transcription initiation is then under control of the E. coli promoter. The chimeric plasmid, which is the plasmid vector with the insulin gene now attached, is then placed back into the E. coli cell for messenger RNA to occur, usually inside of a fermenter at 30 degrees Celsius where translation occurs. The insulin is then harvested and purified for genetic modification to reduce insulin degradation by a citriconylation reaction. Insulin is then made available as an injectable, which is usable by the person suffering diabetes when glucose levels are too high. Injections are usually made into the abdomen as this is the quickest route. Next, let's take a look at the human papillomavirus, or HPV. It is a sexually transmitted virus, often with no immediate symptoms or obvious signs. The typical strand 16 and 18 are the main causes of cervical cancer. HPV is a double-stranded DNA genome virus which attaches onto the mucosal or cutaneous epithelial with icosahedral capsid or shell around the gene. The recombinant protein for HPV is a vaccine and falls in both the Group 2 therapeutic proteins with special targeting activity for interference with molecular organisms, and also in Group 3 for therapeutic proteins as a vaccine. The vaccine for HPV is produced by adding an L1 capsid protein virus-like particle, or a VLP, of HPV type 6, 11, 16 and 18, which is also expressed as Saccharomyces cerevisiae into an eukaryote yeast gene plasmid known as a baculovirus expression vector. This forms an empty shell that resembles the HPV virus. After the transcription process, virus-like particles are purified by removing impurities by a series of chromatography. An adjuvant of aluminium hydroxyphosphate sulfate then absorbs the VLPs, which will enhance the body's immune system antibody production. The final VLPs are then injected into the deltoid muscle to begin mimicking the HP virus and creating said antibodies to destroy the virus should the immune system ever encounter it. It is also important to note that the vaccine for HPV has no viral DNA, so therefore cannot duplicate or infect the cells. The vaccine was developed in 2002. So what does the future look like for recombinant protein? Looking back at the categories outlined by Leder, Bakker and Golan, we can break down each category's projected future applications. For Category 1, therapeutic proteins with enzymatic or regulatory activity, as the medical world discover more protein deficiencies of different variations throughout the body that link to certain diseases, the need for new treatments will increase drastically. For Category 2, therapeutic proteins with special targeting activity, Leder, Bakker and Golan 2008 state will probably expand further as signalling pathways and etiologies of disease are more clearly identified. Just think about the groundwork that is being put in to find a cure for cancer. One day, and hopefully soon, we will have a breakthrough in that. Also, as we start to understand that the selectiveness of the blood-brain barrier is hard to cross with recombinant proteins, there are laboratories working on proteins that will combine with other proteins to allow this delicate crossing to enable a treatment for neurological disorders such as Parkinson's disease or even Alzheimer's disease. For Category 3, therapeutic proteins as vaccines, this category undoubtedly will consistently be evolving as new viruses are involved and exposed to the human population. It will include cancer vaccines, influenza strains, and a tirade of infectious disease. And lastly, for Category 4, the therapeutic proteins in diagnostic, clinical and research-based proteins will continually be adjusted and tested to create recombinant proteins needed for vaccines or proteins to work alongside other proteins, as in the case for the blood-brain barrier crossing. and every other new illness or disease in which the medical world can alter, cure, or vaccinate against. It is an exciting time to be witnessing the ever new and evolving world of science.